Good morning, everybody. Welcome to today's webinar, where people are signing in now as we have just launched today's webinar. My name is Kerry Oceans. I'll be your moderator for today, and I'm just going to wait another minute or two while people get logged in. We've had a very strong response to today's webinar. Over 400 people registered, and we expect at least half of them will actually be listening in today, and those that didn't will have a chance to listen to the recording at a later date. Today's webinar is brought to you by the Composting Council Research and Education Foundation, CCREF, and BSI Bio. The funding for the development of this program has also come from the Biodegradable Products Institute, BPI, and the Food Packaging Institute, FPI, and so we thank both of them for their support. For those of you not familiar with the CCREF, this is a little bit about us, our mission statement. You can see some of the things that our work centers around, including scholarships and research. We provide scholarships for high school and college students interested in investigating composting and compost use. We've trained over 1,000 people in our compost operations training courses. Our toolkits and online resources is an area we're looking to grow, and we manage the annual International Compost Awareness Week, and you can see the winner of this year's poster contest over there on the right. The program is also supported by the Composting Council itself, our sister organization. Many of you on the call are already members of USCC or members and participants in our STA certified compost program, and maybe registered for our upcoming conference in Charleston, January 28th to 31st. For those of you who aren't registered, we're going to offer an 11% discount because today is December 11th, and that'll be good for the next 11 days. You'll get an email about this tomorrow, so please take advantage of that. Finally, I'd like to go over a few housekeeping notes for today's webinar. Everyone is starting on mute to maintain order. You'll see in the attendee control panel that if the sound quality isn't good on the computer and you want to try calling in, there'll be instructions on how to call in by telephone. You'll have opportunities to submit text questions to today's presenters by typing into the question panel on your control panel. You may send any questions anytime during the webinar. I will be collecting those and we'll address as many as we can during the Q&A session at the end of today's presentations. And we understand that there are lots of questions about compostables and contamination and the relationship between the two. There may be questions about ASTM standards, about separation techniques, technologies, about ride-along contaminants like PFAS, certainly a growing area of concern. But we will not be addressing those questions today. That is not the focus of today's webinar. So I'll only be able to ask and get answers to questions on the field testing program itself, the subject of today's webinar. And finally, today's webinar is being recorded. Tomorrow you will receive an email with a link to the recordings. And so now getting into the meat of the program, here's our speakers for today. We have Emily McGill, who's from BSI Bio, and she'll be describing the evolution of this field testing program and what's involved today. Laura from Calgary and Marissa and Charlie from New York will be following Emily with their experiences participating in the beta testing of these protocols. And finally, Wayne Howard will be here to wrap up with how you can participate in the future. And I'll be providing a little more background of each of these speakers as we transition from one speaker to another. With that, I'm going to stop sharing my screen and let Emily bring up hers. And while she's doing that, let me let you know that Emily is the manager of research and communications for BSI Bio. BSI Bio Packaging Solutions, and is a founding partner in the compostable field testing program. Emily's areas of expertise are in waste prevention and resource recovery, especially at the intersection of food and waste. She has designed and managed field testing with compost manufacturers, and she's co-founded and facilitates the Master Recycling of Vancouver, an adult education program for waste prevention and reduction. But Emily has a bachelor in bioresource engineering, and she brings a passion for integrating research, analysis, and design for systems change. And she's been a great partner in the development of this program. And with that, Emily, go ahead.
Great, thank you, Carrie, and welcome everyone. I'm so excited to be having this webinar. It's been a long time in coming. Thanks so much for taking the time out of your day. So I'm the first presenter because I've probably got the most context on the origin and operation of our program. It's called the International Compostables Field Testing Program, or the CFTP. So we'll be using that acronym scattered throughout today. As Carrie mentioned, I work for BSI Bio, and the topics that I will cover today, if I can switch my slide, is a little about our company and our role in developing the program and operating it today. I'll go over the distinctions between certification for compostability and field testing. I'll talk about the development, the origins of the program, what testing looks like today, and then there's a couple quick frequently asked questions that I'll cover. Jumping right in. BSI Bio is based in Vancouver, British Columbia. We were founded in 2005. We design and distribute compostable foodware. We've been field testing since 2014 because over the first 5 to 6 years of the company, our CEO, Susanna Carson, learned that through a survey that in the Pacific Northwest, only about 50% of facilities were considering accepting compostable plastics, even if they were certified. A quarter were rejecting outright, and a quarter were sitting on the fence. She brought me in to design and implement a field testing program just for our products, doing academic research with local facilities. It was this early research that led us to the program as it is today. And right now, BSI Bio and the Compost Council Research and Education Foundation are the two co-founders. And myself and Wayne, who will speak later, are the two program coordinators who help facilities navigate the testing program. So, this program is really meant to be addressing the issue that these certified products are getting into our stores, our restaurants, and even though they're certified, they're not facility accepted. Hence the field testing program, how we got to here. The next two slides are really text heavy, providing you just a sense of the timeline and the amount of work that has gone into its development. In 2012 to 2013, the US Composting Council worked with Cascadia Consulting in the city of San Jose, California, doing field testing with the intent to develop a shareable test method for the mesh bag disintegration process for compostable products. The result of that work was indeed a test method that they shared open source at the same time that I was hired and started looking at the literature to see who's doing testing, where are they doing it, can we borrow their process and implement it where we are. From 2013 through to 2015, we worked with US Composting Council, the folks from Cascadia who'd done the original testing, with a lot of incredible advice from other consultants in the field, industry experts, facilities themselves, and facilities that were already doing testing to adapt those protocols. And up in Canada, we have a government body called NSERC, National Natural Science and Engineering Research Council, that funded a 6-month study with us for the University of British Columbia, where we created an academic study with a postdoctorate researcher to get this method integrating all of the technical advice that had been received during the San Jose development. which of course resulted in a formal partnership between the Compost Council Research and Education Foundation and BSI Bio in 2016. So since 2016, over the past 2.5 years, we've been doing beta testing with a variety of facilities at different scales and in different locations across North America. And with assistance from the Food Packaging Institute and Biodegradable Products Institute to fund, we developed an online data collection system. And now, partly with this webinar, and of course, in the coming months with some more publicity around the program, we are in alpha launch, inviting any and all compost manufacturers to join into this data collection program. I'll do a quick nod to some early publications that you can refer to as well. So on the left, you have the academic research that resulted from that early study with UBC, and then an announcement, a sequence of announcements in BioCycle when we first launched. Where's the program going from here? After 2022, we're going to continue alpha testing and refine our data collection system. There's still a few kinks to work out. Once we have enough data collected and enough facilities have participated, we're going to be anonymizing and sharing the results. And then beyond that, we're planning to adapt and improve the program and work with other institutions, media outlets to publicize the correlation between different material types with different facilities and operating conditions. So just in brief, I do want to mention that the compostable field testing program is not the only field testing program out there right now. There's plenty of others, including for-profit, private consulting, facilities who've been testing for many years and continue to do so. About this particular program though, some of our values are that we're nonprofit and open source. We're doing this so that the results can be shared as readily and as widely and openly as possible, and that it's digestible for any researcher to come along and interpret and build on the results. It's multi-stakeholder, involving folks from both the compostable products industry and the compostable manufacturing industry, as well as multiple industry associations, and it is international, spanning Canada, the US, and we're definitely fielding inquiries from all around the world because this is a need for any growing organics industry. Also in this program, we have standard materials that are tested in all facilities. It's customizable to multiple sizes and scales, and we're anonymized by default. What that means is that facilities who participate in the program can opt to have the name of their facility shared with their results, but by default, it will not be. Moving on to certification versus field testing, I'm going to be covering topics that I think most people on the webinar will be quite familiar with, but for the sake of the recording, I'd like to cover a few basics. One is a quick terminology check. The real purpose of certification is to be able to distinguish between these different material types. The thing I want to point out here is that bio-based has nothing to do with whether something's biodegradable or compostable, and that all types of degradation, oxo, photo, the X, the mystery degradable, and biodegradable are not rigorously certifiable terms at this point, and only compostable is rigorously certifiable, and it means that it's going to biodegrade under specific conditions, producing usable compost. Certified compostable means that an item can be biodegradable but not compostable, but all compostable products will biodegrade under the right conditions. Takeaways: bio-based materials may or may not biodegrade, biodegradable materials may or may not be bio-based, and neither term means that they're compostable, though they could be. Certification helps make this clear. If something is not certified compostable, you can't know for certain that it's going to break down in the set conditions. When I say break down, there's also a couple different ways to look at this. The first is disintegration or fragmentation, which really just means breaking into smaller and smaller and smaller pieces, but maintaining the same type of characteristic. You can imagine a Lego house that you take apart and you have individual Lego pieces, which are still fundamentally the same material. Biodegradation or composting or decomposing is a little different. It means disintegrating and fragmenting, yes, but when it gets small enough, those Lego pieces actually break down into their individual molecules and get transformed into other products. So fully biodegraded items don't retain their characteristics. This is the goal of having compostable products end up in facilities, but as we know, this is not happening everywhere. So the key distinctions and really the purpose for the field testing program is not to replace certification, but to expand on it. Certification is very important because it assesses both the ingredients and the disintegration level. So the lab test shows that the item will biodegrade within a set time period, but it also checks that there will be no toxic residuals. Whereas field testing in real world conditions just takes those lab-based results, assesses them, and relies actually on having the products be certified compostable so that you know you're not contaminating the compost at a microscopic level. I'm going to speed up a little bit because I want to leave time for the other presenters, so bear with me. This is what today's field testing looks like. There are basically 5 program stages: planning, preparing, running the test, assessing disintegration, and reflecting on results. In the planning stage, which you could start even today, you connect with us over website or email, review the terms and conditions of the program, including the anonymity of the data, and we'll tailor the test plan via phone calls with you. During the preparation stage, you'll receive the test package which we provide, engage your test team at the facility, and gain online access, which will walk you through for how you're going to collect the data. When you're running the test, you have to pack and load the test bags, manage the intervals. So whether you're turning the pile or moving from, say, a static pile to a windrow, that's what an interval would look like and how you're going to manage the bags in between. And then you'll remove the bags before you screen or before you cure. It's different for every facility, and this is why you have to finalize and refine your test plan during the preparation stage. Once you've removed the bags, you screen them. We'll work with you to determine what size of screen you want to use. We do have standardized recommendations. You pull out the product residuals, you weigh and report, which of course leads to the reflection stage. You'll review your results, how much are the products degrading or not, and decide on your next steps. What we really like to see is if one of those next steps includes repeated testing. Whenever you do a field trial, you're really getting a snapshot of how products degrade with the given feedstock at that given time of year. You could test again in a new season, maybe with a new location or a new facility, or even in a different type of process within the same facility, or you'll want to test additional or new products. So, to stave off a few of the questions that I think will come later, here are a couple quick frequently asked questions. What comes in the test package that we send you? What materials are tested? Can you customize the products? And do we have a method for having in-vessel dose tests? First, the test package contents. There are standardized sample products that are tested across all facilities, mesh bags to put them in, along with feedstock. We provide labeling ties to distinguish between bags, rope so that you can pull the bags easily in and out of piles and identify where they are. And then, of course, loading instructions, which again, we go over in real time with you as well. The standard materials that are tested across all facilities fall into 3 categories. There's fibers, polymers, and then fiber polymer laminates. I've listed here a sample of what the actual products are that are being included. What's been selected are the larger formats of some of these products, because if you test a larger product that has the exact same material makeup as a smaller one, and it breaks down effectively, then you can know that that smaller one, which is easier to degrade, is also going to break down. What's been chosen are a good example of what you might get in a typical food court that's gone 100% compostable, but it is not all exhaustive. And I'm more than happy to discuss this at length in a follow-up. Can you customize your test products? Yes, absolutely. All that we ask is that you definitely maintain that same standard test bag that we provide for you so that we do have that replication of the same products across all facility types, which is going to help us draw some correlations between operating conditions and disintegration. But we'd love to support you in testing something that is specific for your locality. We select this during the planning stage. You'll source the custom items. We help you figure out how many bags you'll need to test the number of items you want, and then we'll provide those materials for you. Facilities who've tested with us that have added custom items often will add straws, multi-laminate bags like coffee bags or the stand-up pouches you get in grocery stores, alternate types of fibers like a wheat fiber, and specific cutlery brands. Cutlery is where there's the largest variation in terms of the actual ingredients, and it's important if you have a different cutlery brand than the one we provide that you might want to test it as well. Lastly, do we have a method for testing in aerobic in-vessel facilities? This is still in the works. What's involved is a massive amount of products not kept in bags, put into an entire section of an in-vessel facility, and then you have to subsample at the other end. We've done one section dose test to date, which is not enough to say that we have a replicable protocol. We would love to invite additional partners to develop and validate this method with us. You are invited to sign up at compostfoundation.org/field testing, or you can email me directly at compost@bsibio.com. Thank you so much.
Thanks. Thank you very much, Emily. I think we've got now 270 people who have logged in. And I think before we start Laura's, our next section, which is some reports from the field that people have done the testing, the beta testing. I've got a quick poll for everyone. And so you're seeing in front of you just a quick question about why are you here? And so this will give us a sense. We know from the pre-registration who was signed up, but I think we thought it'd be interesting to see actually who would be here. So we've got about a third of you have voted so far. This is awesome. Thank you so much. We're going to get another couple of seconds here. All right, we're at 3/4 of you have voted. All right, looks like it's slowed down. So the rest of you probably just aren't paying attention. We're going to end the polling now. It's been 1 minute and we'll share the results. And as you can see, so it's pretty evenly split between the composters and researchers and educators, and that is just awesome. You're the folks we really were hoping to be here. We're happy to have the other folks here, the product manufacturers, distributors, and those of you who are just interested. That's awesome. And with that, I'm now going to have Laura Hamilton. Laura, if you want to launch your, share your screen. And while you're doing that, we'll just do a quick introduction for you. Laura is the waste diversion specialist with the City of Calgary in Alberta, Canada. She manages the municipality's Green Card program, working closely with the largest in-vessel composting facility in North America. Her role includes regular monitoring of the industry, composting trends, education and programming service for the residents, and planning annual compost giveaway events for the thousands of attendees. Laura has also worked as a waste educator, working with businesses and organizations to share best practices for implementing diversion programs. Prior to joining the City of Calgary, she worked as an operations manager at a startup composting facility, developing processes to ensure worker safety, regulatory compliance, and compost quality. And go ahead, Laura.
Hey, thanks, Carrie. I'm with the City of Calgary up in Canada, and what you're seeing is an overview of our facility that we opened in 2017, so it's a fairly new program. And so today I'll give a quick overview of our facility for context and some of the reasons why we participated in the testing program, and a few slides on our experience going through the testing protocol and what our next steps were, and a few tips for success if for anybody who's thinking of participating in the future. So as Carrie mentioned, we have an in-vessel aerated static pile system, and it is one of the largest of its kind in North America. It is right within our city limits, so we needed it to be contained and indoors for odor control, and we have over 500,000 square feet, including 18 vessels for active composting, curing area plus storage. And we process over 145,000 metric tons of SSO and dewatered biosolids each year. The majority of that is the SSO. And we are a fairly new facility, and so we've rolled out the program with Green Cart serving over 330,000 households in the city. And so we've had just over 2 years of operating now. So from the start, we have accepted all food and yard waste and pet waste as well. And we do accept the certified compostable bags. But aside from that, we haven't accepted any other certified compostable materials. So this is a quick overview of our process. So our biosolids and SSO streams are kept separate, separate vessels, and marketed as distinct compost products. This test was only conducted in our SSO streams, so the Green Cart materials that are food and yard waste. So when material reaches our facility, it goes to the tipping floor, goes through a shredder, that will break open bags, yard waste bags, large materials, and then they are moved into one of 18 vessels for 21 days. And after that 21-day active composting period, materials goes through screening and then on to curing for another 21 days. So, a few pictures of our receiving area. All of our municipal trucks come in and dump the food and yard waste. And the shredder is on the bottom corner. And then it moves into our composting vessels. So these are very large and with aeration in the floor. And we use a lot of large equipment to move materials around. And we'll go into screening and then finally curing. So some of the reasons we decided to participate in this test was because, well, the ASTM standard is up to 180 days for decomposition, whereas our process is quite fast. It's less than 45 days. And we screen after 21 days. So we need things to move through our facility fairly quickly to keep up with the volume. And so we wanted to, and we didn't include other compostable products in our original list when we rolled out. So we wanted to be fairly conservative and then be certain of our process before we expanded that list. And of course, there has been a lot of increased interest from the public and local businesses as zero waste movements, issues with single-use plastics come along. And we also wanted a deeper understanding of how our products were performing in our facility compared to the testing required for certification. And a better understanding of what's out on the market. We were a new facility and just getting a feel for what's out there. So this is a good exercise for that. And some backup for our program decisions. If we don't accept materials, it's good to have some data to back up those decisions. And bigger picture, hopefully we can contribute to some advancements in the industry and better collaboration between manufacturers of these compostable products and the processors who are expected to receive them. Oh, sorry, I'm not sure what's going on with my slides here. Okay. So just gonna have to one moment here, sorry.
It's okay. Take your time. Get it set.
I'll be right back. I just gotta, I think I'm on Hopefully this got us off of the. All right, so, sorry about that. Oh, it's still happening.
Laura, your sound is a little weak also. It was working better before.
Okay, I'm not sure what's going on here.
Why is my face showing up?
Really sorry about this. I'm not sure what's going on here. So, all right. So I guess I'll so this is loading the bags. We've simulated the materials going through the whole phase of the our system. So transportation with other wet organics. So we dip things in the water, we crushed them and tore a few things to simulate it going through the shredder. And then we layered with the organics within the mesh bag. So this was late fall, so there was quite a lot of yard waste in there at the time. So loading vessel, this took a little trial and error to figure out what would work best. We had our loader create a bit of a shelf in the vessel after it was most of the way full of organics. And then we tied all our bags together to a rope with lots of flagging tape so we could recover them later on. And then that load of organics was placed on top of the bags. And that was about 6 feet up in the air, so we could try to get it as close to the center as possible. So after 21 days, the bags were removed again very carefully with the loader and some shovels to remove them by hand. And then we placed these bags in buckets so we didn't lose any material through the holes in the bags while we were waiting for them to dry. And so after drying for several weeks, we opened them up over a multi-level screener. And some of the materials had decomposed so completely that we didn't find anything at all, or just a few fragments. And the two pictures you see up here, the one on top is just the shadow of a coffee cup lid. And as soon as we touched it, it just kind of disintegrated into dust. And similar for the other picture, which was a spoon. So that they did break down. We did have some variability between vessels and test bags, and we did have other materials that we used, we sourced locally, and we did find some mixed results among different brands compared to the standard set of materials provided. So next steps for us is uploading to the database. And this summer, we also used this test as a baseline and protocol as a framework to conduct 2 larger scale bulk tests using material from a local festival. They did a really good job of sorting and controlling what products were available for this festival. So it was a fairly clean source of materials. And so we put those through all phases of our process that weren't part of the initial mesh bag test. So they went through receiving, shredding, screening, and then finally curing as well. And so we're still compiling all the results, and we'll review any potential program changes if they make sense. So for anybody who's thinking of doing this, participating in this, you can expect to spend about 2 hours a bag with 2 to 3 people, including preparation of the bags, loading, weighing, measuring, placement of them, recovery, and analysis. And of course, that will depend on your particular facility and setup and process. And it's good to have the same group of people working on it throughout to ensure consistency and documentation and what to look for in the process. We did find that some of our materials were quite compacted after they had been dried. So if you can break things up before they go into drying and monitor that every few days, that could help a bit. And definitely be sure everybody at the facility knows what's going on, and to not disturb any of your testing materials or anything that is waiting for you to analyze. So these don't walk away. Oops. And finally, take lots of notes and on your conditions, the feedstock, anything that may have influenced your results. We did have one test bag that broke off the rope and had migrated up towards the top of the pile. So the results were a little bit different for that one, but so make note of that, and then you can have some explanation if you do have outliers in your data collection. And that's it for me. I'm sorry about the slides racing ahead there. Not sure what happened there.
That's okay. Thanks, Laura. Thanks for persevering. Yeah, you did a lot of, that's a lot of work that goes into those, into the testing. So I think hopefully, so folks will have some questions specific to the testing. I am seeing some of the questions being asked are kind of off topic. And so we will probably not be getting to those. Again, would like to stay focused on the testing, field testing program and protocols. And so with that, Marissa the Dominicis is with us, along with Charlie Bear from Earth Matter. If you two want to start your screen share, I will be introducing you. First, just a little bit about Earth Matter itself. Earth Matter is a not-for-profit located on Governor's Island in New York City Harbor. It's a fantastic location if you ever get to go. Earth Matter is working in partnership with the Trust for Governor's Island to develop a zero-waste island, and the Trust mandates use of compostable serviceware by vendors and event producers. This year, with the generous support of the Department of Sanitation from New York City, they processed over 1.3 million pounds of food scraps, compostables, and yard waste generated on the island and with some neighboring food scrap drop-offs included at New York City farmers markets. Marissa is Executive Director and co-founder of Earth Matter New York. Prior to that, she worked for 18 years as an environmental educator for the Trust for Public Lands. And it was there where she really developed the keen awareness of the need to protect the commons, the green spaces that can be used to build a green culture. Marissa heads up the Demonstration Farms training program and works at the Compost Learning Center, engaging individuals and groups in composting. She's accompanied with Charlie Bear. Charlie is also a co-founder of Earth Matter and the operations manager. Charlie has long recognized the opportunity to improve urban soil health and reduce waste by creating compost locally, and the value in helping others to do the same. His work assures that everything and everyone that comes through the facility gate is transformed, materials into compost, and people into practitioners and advocates of improved resource management. All right, Marissa, can you start your slide share?
Host has stopped it. Hello?
Yes, Marissa.
It's having a hard time uploading. It says the host has stopped the upload.
Try this again.
Okay.
You were here? Okay. Everybody can see that first slide?
Yes.
Great.
Oh,
Okay.
it just disappeared. It was there for a second.
Is it there now?
Yes.
Awesome.
Yep.
Thank you, Carrie. Like you mentioned, our Compost Learning Center is our home base, and we're located along with other environmental and urban ag collaborators in the urban farm on Governor's Island. We have 4 program areas: our Zero Waste Island Initiative, our New York City Compost Project, educational and community compost scale operations, our Soil Start Farm, where we demonstrate on-farm composting methods and application in growing food, and our Lavender Field is a place that we designed to evoke a sense of beauty and healing. Our educational mission promotes experimentation. We feel we're learning from people, we're learning from plants and from the soil, and this makes our center a useful place to trial materials and systems.
The urban farm that Marissa spoke about utilizes the open spaces within a neighborhood of vacated townhouses that were family housing in the period when the Coast Guard occupied the island. Our compost pads are former parking lots, and the farming activities take place on what used to be lawns.
Excuse me, Charlie. We are only seeing your picture of a chicken. We're not actually seeing your slides.
Uh-huh.
I think, are you showing your PowerPoint slides? Yeah, that's what you have to shoot when you choose share. You have to share your slides, your PowerPoint.
Okay, so we're back up here?
No.
Mm.
Check the share button and make sure you see share my screen. I'll tell you what, I will share I will share my screen. I have your slides, and then I think that's, you can hopefully you'll see them, and then we can proceed that way. Let me.
That sounds good, Carrie. Maybe you can tell us, we'll tell you when to advance it.
Yep. Thanks everyone for holding on with us. So share.
Okay, as I was saying, the urban farm that Marissa spoke about utilizes the open spaces between and within a neighborhood of vacated townhouses that were family housing in the period that the Coast Guard occupied the island. Our compost pads are former parking lots, and the farming activities take place on what used to be lawn areas. The Compost Learning Center is a New York City registered compost facility. That status allows acceptance of up to 10,000 cubic yards of yard trimmings and 2,500 wet tons of SSO per year. We're currently accepting 300 to 400 cubic yards of yard trimmings that are generated on Governor's Island. And as Carrie said, about 350 wet tons of SSO, principally from residential drop-off locations at farmers markets throughout the city. It's very clean material. I'd like to focus on first on how a small facility like ours, without a grinder, a mixer, or sophisticated screening, can process compostable products. The slide in view is the receiving, tipping, and blending area, a typical Monday morning with 90 or 100 bins and 20 or 30 toters arrayed around our base of wood chip. Sometimes the base is yard waste. It's 6 or 8 tons of material ready to be tipped out. The thing I'd like to call your attention to is the center rear bag, pile of green bags containing the compostable products from a weekend event. And also the foreground of the picture, the 2 corners, is the previous week's material, where all of that incorporated compostable material has already principally disappeared. We can advance the slide. Principally operated mechanically aerated static piles, initially advised by Peter Moon of O2 Compost. We made that choice to limit the labor effort, the machine time, and the potential for odor generation. Low odor, neighbor-friendly compost is essential for a site that is open to visitors each weekend and has no perimeter buffer zone. For 6 years, we operated varied configurations of the ASP and expanded ASP with organic compost covers. Last year, we had the opportunity to trial the sustainable generation SG Solar, powering the SG mobile system with a gore cover. We could advance the slide. We're now using that covered in-vessel system to increase our capacity, retain moisture, reduce labor, and improve the site aesthetics for the future. A movable system like this is essential to us. Our facility will be relocated when future development occurs. And for that reason, both of the systems are solar powered. We retain the O2 system for the curing and maturing of material. Retention times in the early stages are 4 to 6 weeks with extended periods for the older material. Forced aeration is maintained right through to screening, and total process time is 12 to 15 months. Slide advance.
Okay, so this is the Compost Learning Center, a hoop house, and some of you listeners may be in this photo. The Community Compost Forum in 2019. Thanks to the great organizing work of the Institute of the Local Self-Reliance, we had 80 community composters come from around the country for a workday. At Earth Matter, we feel the key to learning is participation. All of our activities are open to the public. We offer Monday community compost builds, Friday work days, and our intensive farm apprenticeship program and weekend public drop-in hours from May until October. Next slide, please, Carrie. A major component, the zero waste island. While the habit of using single-use disposable items is not the direction we want society to go, we feel we cannot ignore our culture's current trend. In 2018, the Trust for Governors Island became a formal partner of the Zero Waste Island Initiative, which we call ZWE. The island is open, and this year they hosted 750,000 visitors. ZWE encompasses education, consultation, transportation, and processing of visitor, vendor, tenant, and event organics, compost to be used primarily on the island's landscape. Our ZWE team, on the left, collaborates with all partners on Governor's Island. The team consults, manages, and provides educational training for the maintenance staff, all of the vendors, event producers, and this team handled 76,000 pounds of SSO, making sure our compost project operators had clean, usable inputs. Based on the heightened visibility of the Zero Waste Island and the attractiveness of the island, we expect this volume will double in 2020. And in 2020, we're looking forward to a grant, using a grant that we got from the New York State Pollution Prevention Institute, piloting a compostables buyer co-op to help with reducing the costs and increasing the availability of BPI certified materials for island partners.
Next slide. Now returning to the receiving area, we see that while Marissa's been talking, the bins and the toters have all been emptied onto the wood chip as a second layer. The compostable bags are beginning to be emptied as a third layer. That lasagna will be inoculated with a thin layer of active compost and topped with additional wood chip. Bucket blending along the long axis of the pile mixes the materials, and a cover of leaves and straw will cap that windrow. Very little of the compostable product escapes this process, although we do have some special techniques for incorporation on especially windy days. Compostable products generally make up 2 to 5% of the pile by weight. Our initial moisture content is 50 to 60%, and CNN is estimated at 30 to 1 or better, with the initial temperature range in the in-vessel system being 170 to 180 degrees. With the organic cover, formerly it was a little bit lower in the 150, 160 degree range. And we find that products disintegrate in 7 to 84 days, 1 to 12 weeks, depending on moisture and temperature parameters. And as previously mentioned, the ambient conditions. The rare scraps that do make it through to screening are regarded as overs to be recycled, not as contamination. That said, clearly screening does concentrate what true contamination there is, eventually making the overs unusable. Next slide. And there we have the full, pretty much the full volume of the compostables overlayering the top of the pile. Next slide. Our trial of the protocols took place in early 2017 at a moment when our work had shifted temporarily to a portion of the site that did not have electricity. So these bags were incorporated into a turned windrow. And unfortunately, that windrow responded in a way that at the time we described simply as enigmatic. The temperature arc wasn't like anything we'd ever seen before. In short, the test just didn't get off to a good start. We continued to include the bags in each phase of the pile, which eventually did reheat. Removing and replacing the bags did become something of a chore. It was a little difficult to maintain our enthusiasm because our prior experience told us that the types of products in the bags would certainly be decomposing in our piles. So the data story of that pile remained untypical throughout, and it's really likely not replicable. That said, the materials all did break down completely. Next slide. Yes, this is a shot of the bags partway through the trial. And as you can see, the bags themselves do show some wear from the continued removal and reinsertion. Next slide. We have gone on to use the protocol for a trial of this coffee bag. Our neighbors that drop off at the farmers markets often challenge us with interesting interpretations of what they consider to be compostable. And we actually had communication with the woman who dropped these bags off and wanted to assure her whether or not they actually were compostable. It's a heavy gauge bag with an integrated vacuum valve. The residuals on the right are after a full 180 days of processing in our aerated static system. And we concluded that the coffee bag would have completely disintegrated directly if it had been incorporated directly in the pile and been subject to the agitation of moving the piles. So we now accept this bag without any issues. Next slide.
Oh, this is mine. The Zero Waste Island team, with help from our farmers. This is an adaptation of the test protocol. Our ZWE team adapted these protocols by performing the test instead of an industrial setting in a home scale device. In the middle, you'll see the 2 cubic yard batch of compost. We loaded the bag with the polymer, the PLA cups, cutlery, and bowl, and fiber products using the protocol of 20% product to 80% active compost. We incorporated the compostable products that we received from our vendors. We monitored the sample bag after we placed it in the middle of the pile, and we followed the thermophilic PFRP guidelines of turning this pile 5 times in 15 days, and we maintained the temperatures above 131 degrees in all parts of the pile. Our results were that all the compostable materials in the sample bag reached a level of successful disintegration within 12 weeks of retention.
Next slide. So in conclusion, I'd like to say that we do regard the protocol as a valuable tool for testing new and unrecognized products. The open sharing of the database over multiple data points of CN and moisture and temperature turning intervals should be a really illuminating practice for everyone who participates. And this last shot is of the creative and dedicated team of New Yorkers that is questioning the status quo of waste disposal. And we're open to all of your questions as well. Thank you, Carrie.
Okay. Thank you, Charlie and Marissa, and thanks for adapting. You know, you practice these things, and you have to have backup plans. So, last up, we've got Wayne Howard. Wayne is program manager for the CCREF. His duties include coordinating the Foundation's compost operations training program, oversight of the compostables field testing program that we're talking about today, and development of educational tools such as compost sampling videos. Prior to joining the Foundation, Wayne worked as a review engineer with the North Carolina Division of Water Resources, evaluating municipal and community water systems, water supply planning programs. He holds a master's in biological engineering from NC State University. Go ahead, Wayne.
Hello, everybody. Mine's pretty short and sweet here. So you've heard a little bit about the program and heard some trial and errors we've had. And so if you're interested in joining the program or just finding out a little bit more, the best thing to do is head over to the Composting Council Research and Education Foundation website. If you kind of look in the upper corner of the screen, the website address is www.compostfoundation, all one word, .org. And then from there, under the research tab, you scroll over to research, just go down to field testing, and then we can, and that takes you to our International Compostables Field Testing Program. Conversely, you can also just type after compostfoundation.org, just /field testing, all one word again, and that will take you directly there if you don't want to search through all the menus. And, but once here, as you see, you scroll down, we have a little basic information, very basic, about the program itself. Then we have an interest form, excuse me. And so you just fill out your information here, just first and last name, your organization, if that applies, best your contact information, and then your reason why you're here to contact us, you know, if you're interested in participating or just have more questions. As mentioned previously, we've touched on it, participation is anonymous, and once a sufficient amount of data has been collected from what we consider a substantial test group, the data will be anonymized, sorry about that, and made available to the public. There is no cost to participate. However, if a facility is being paid by a vendor or manufacturer to participate, then the expectation is a percentage of that payment would go back to the program to help further research. And just to scroll down here, as we've mentioned before, you know, CCREF would like to thank our partner, BSI Bio, for all their efforts in making this program a reality. Also, a huge thank you to the Biodegradable Products Institute and the Food Service Packaging Institute for their generous project funding that they have provided that's gotten us to this point. We appreciate the time and energy spent by our program's early adopters who've helped us to refine our testing and data collection processes. And lastly, we'd like to thank you, our attendees today, for your interest in the Compostables Field Testing Program, and we look forward to working with many of you in the very near future.
Okay. Thank you, Wayne. And yes, we do encourage folks to get in touch with us to participate. Let me just sorry about that. Okay, it's time for question and answers. We've got quite a number to ask about. I appreciate the ones that have come in. So I think Emily, make sure you're off mute. Right. Unmute. Emily, this one is for you. is from David Fridlin. He asks, first, thanks for putting together this program. Would you view and actually this is for any of the compost producers as well. Would you view the sample bag as a representative sample of the entire category? In other words, when you test a basics molded fiber bowl and it passes, that is, it decomposes, would you then be comfortable extrapolating that to the acceptance of all other fiber bowls?
Question, David. And this is something that is a conversation with every facility, because it really, the program is not intended to replace certification or to be the only way that a facility will assess acceptance. But we do advise that for specifically the fibers that are very common, the fiber polymer laminates that are very common, and any of the pure polymers that are extremely common across brands, that they could extrapolate those results. There are some particular product areas where we would not recommend extrapolation because there is such a huge variation. So as I mentioned before, cutlery is one of those product types where it's representative of maybe half of the cutlery on the market, but they might want to include some other brands for that particular product line. I'd love to hear, though, what Laura, Marissa, or Charlie would speak to this as well.
Sorry, I couldn't hear that last bit, Emily.
Would you, for yourself, consider that the results you saw with the standard product pack that we sent you, would you extrapolate the results for those products to represent the different compostable foodware that you get in your existing feedstock from your local market?
No, I wouldn't. Based on the results we had from doing we had the standard set of bags and the some locally sourced materials, there was quite a bit of variability, and that also was reflected in our bulk testing that we did this summer. So it's yeah, there was it's a starting point, I guess, and then yeah, we did find some variability, so I wouldn't say that it's universal.
And I would agree with that, particularly in the product line of cutlery. I think that you're right, that probably has the greatest variability.
Agreed, yeah. We found a lot of difference with cutlery especially as well.
Great, thank you. Question from Peter Adrian, asking about how the test is able to simulate the effect of turning and shredding and aerating that the typical, in a typical windrow versus what's going on in the mesh bags. So I guess the question is how do you accommodate for that, or how do you account for the mechanical effect of turning and that doesn't happen in the mesh bags?
It is a great question, and this is the #1 flaw with this method of testing. Unfortunately, there is no way presently that we've built into the program to account for that. So when you reach an interval where you will be turning the windrow and aerating it, the bags are just removed, kept warm, and then replaced back into the pile. So this is why many facilities, and as Laura even mentioned herself, will follow up the mesh bag testing with the kind of dose method where they will actually identify a section of their pile or windrow where they will provide an evenly distributed dose of the same products and then assess so that they can get that representation. As mentioned, we don't yet have a very thorough protocol for that type of dosing method. We just require more tests following that so that we can kind of validate the results.
And I think I will add that I think in any case, that creates, because of that difference, the mesh bag test is conservative in that if materials are disintegrating in the mesh bag without that, whatever additional effect of the turning would have, then you would be confident that with that extra turning, it's only going to happen, disintegration will only happen faster, so that the test would be conservative in that aspect. Okay, question from Neil Edgar to Laura. Laura, and the distribution of the test bags was across a very similar elevation in your pile. He says, I've often heard that aeration, temperature, and microbial activity are more often variable along the vertical axis of a pile, and was wondering if that placement was only one of the options, or if there was consideration of the potential variability in performance in different pile locations.
Yeah, so the way we did things was certainly there were limitations. Yeah, there can be variability within the vessel. So we chose that just to sort of strike that balance between making it accessible for us to place them, accessibility for the equipment. So yes, certainly for future testing, we could play around with different locations within the vessel, and that may impact results. But you know, this was our first crack at it, and so we did the best we could, balancing all of those different considerations. And but for future testing, we can certainly consider different locations within the vessel. For our bulk testing, we were more within the middle of the vessel, but we've been keeping track of where things are inside the vessels, and so we can see if future tests that does have an impact.
Okay, thanks, Laura. How are we doing on time here? We got about we're gonna end the Q&A at 11:15, so we got about 8 more minutes. Dr. Ramani Narayan asks, how do you confirm, ensure that the complete biology and biochemistry processes are occurring, the continuum from mesophile to thermophile and back to mesophile? As part of this, do you confirm the humidification process by characterization of the humidification? And I'll go ahead and answer that really that this test is a disintegration test. The reason that it's tested, we're relying on BPI or other lab certifications that also require and test the actual biodegradation by looking at CO2 evolution and making sure that those materials can biologically degrade and that the results are not, and there are no toxic residues. So that's why the test is only, like any test, there's some preconditions to the test. And one of the preconditions for using this disintegration test is that you're only using certified compostable products. Emily, do you have anything to add to that?
Yeah, just confirming what you said, that we try to, the information we gather has been informed by what facilities say is reasonable actually to expect of them. So pH, bulk density, C to N ratio, and then maturity of the compost at the end. And it is up to the facility to share as much or as little of their temperature profiles, but typically we will have frequent temperature monitoring during the pathogen reduction phase where we can confirm that such stages are reached. I'll also mention that we include a craft control material of a known rate of disintegration in the test bag, so that we know that if that craft control has also not degraded, that the process itself, as represented within that bag, is not representative or is not able to biodegrade even natural materials.
Okay, reading down through questions. Unfortunately, there are a number of questions here that are off topic, and I'm just going to ignore for now. Here's a question from, let's see. Well, this is one from Janice Phillips, which is also off topic, but I'll give a quick answer. She's asking, finished compost with these biodegradable products are not allowed on organic farms. My understanding of these inputs are not allowed. I'm wondering if that's a state restriction or a nationwide rule. And so the quick answer is that's a nationwide rule. It's the USDA's National Organic Program that sets the standards for what feedstocks are allowed or not allowed on into compost to be used on certified organic farms. As of right now, no compostable products are approved for as a compost feedstock. And so, no, the compost that is made using those products cannot be, are not allowed on certified organic farms. There is work being underway to petition or to convince the organic program to allow them, that recognizing that the truly compostable products simply break down to carbon dioxide, water, and become microbial biomass, just the way any other compostable product does, and so should be allowed. But that is the state of the ruling as of now. Richard Tillinger asks, how are you accounting for reduced moisture or air transport caused by the bag being a partial barrier from the rest of the compost?
Another great question, and it relates to how we account for turning during intervals. It is not accounted for during the test. What we do work with facilities on is preparing the bag so that it does model as closely as possible the state of the original feedstock. So for example, with an upcoming test happening at a facility, they thoroughly soak their pile once they've built it. So in this case, when they're actually packaging the sample bags, they will be soaking the mixture of feedstock and foodware before it enters the bag, so that once it gets in the pile, there's no limitation on how much that bag will be permeated when they soak the pile.
Yeah, this is a couple of people probably kind of late, Emily, and are asking about difference between CMA and this testing. I'm going to read you the question, but I think it'd be good repeating the distinction. So I saw Lopez asks, as known, there are different protocols issued by different organizations, CMA in the US, CCC test protocols from the Canadian Compost Council with the same purpose. Any way to have them considered and create one common field testing protocol used by the industry in North America?
Great question, Esol, and thank you for asking it. What I can go over again is that this particular program is nonprofit, open source, and it's free for compost facilities to engage. It's also multi-stakeholder and international, so spanning Canada, the US, and any other countries or international facilities who would like to participate. It's also a little different in that we have these standard materials that are tested across all the facilities. As we know, the variability in a process, even within the same facility between cycles, is massive. So the more that we can limit the variability across these tests, the greater correlations that we can draw in our research. And the other, I think, major difference for this program is that it can be customized everywhere from the home scale testing that you saw Marissa give an example of, up to the size of a facility like Calgary. Lastly, it is anonymous by default, which is a little different than some of the other programs. As for the potential to create a harmonized field testing method, absolutely, there could be some benefit to that. Right now, there is a diversity of offerings because we're still very young in this process of having any kind of standardized field testing. But yes, ideally, there would be something that we could all look to and use across these different programs. I will mention that the three that you've mentioned in your question do follow fairly similar with some slight differentiations to my understanding within the procedures.
Great. Okay, I'm gonna have two more questions. We're going to go over a little bit, but I think both of these will be a good wrap-up questions. First, Dennis Rowe asks, are you monitoring the respiration rate or other parameters during the test trial or at the endpoint?
During the trial, it's not usually feasible for a facility to be assessing respiration if they're not already doing it. If they are, we do invite them to share that information. However, we do ask for all of the facilities to do a maturity test at the end of testing.
And aren't we also asking for temperature and moisture content during the
Yes.
process?
Yes.
Yeah. So So some parameters, some those parameters are being monitored across the test.
Yes.
Okay, lastly, will there be a database that we can refer to get information about compostable products?
Yes, very,
Very important question.
yeah, that is
We're
the
going to end on that one.
yeah, that is the entire point and hope and promise of this program. So right now we have conducted 7 facility tests with data that is was used with the same consistent products and test methods, so it can be anonymized and shared. However, since there's only 7 facilities, we would not be able to maintain the anonymity. So we're aggregating and collecting the data, and we expect that within 2 to 3 years, we will have had enough facilities participate that we can share the database open source. And when we do share that, that's another slight difference between this particular program and the other programs mentioned, is that we will be including specific brands, manufacturers, product composition, some fairly detailed breakdown of the different operating parameters at these facilities, and then the actual percent disintegration rather than just, for example, a pass-fail. So that database will be available online. You can expect it once we've had 20 to 30 facilities participate.
Thank you. Yes, that is an important point of this. And we will have another webinar in a couple of years when we're ready to start sharing those results. And so with that, I want to thank all of our presenters, thank everyone who showed up attended today's webinar. There will be a follow-up email within 24 to 48 hours with a link to a view of the recording of today's webinar. If you have a colleague that was not on here, you can share that link or have them register and they will get their own link. So on behalf of the CCREF, BSI Bio, all our presenters, thank you for joining us and have a great rest of your day.